understand communication with inside your body, and specifically how messages get to cells and create a response. In your textbook, signal transduction is defined as how a message is relayed or passed on to the receiver or responding body. Like most textbooks, this seems a little bit more complicated than it needs to be. All signal transduction is looking at is how a message is detected, transferred to elicit a response that is necessary. The three ways that a cell can respond is either by changing shape or moving the organelles within the cell with the help of the cytoskeleton, to activate or degrade an enzyme to change or manipulate metabolic reactions, or to activate or inhibit protein synthesis. The endocrine system is a system associated with this concept. The endocrine system uses hormones as messengers, and the hormones move through the bloodstream to get to their target cell or organ. There are two types or two characteristics that a particular hormone can have, and that is it can either be an amino acid or protein-based hormone, or it can be a lipid or steroid-based hormone. Hormones themselves are released from glands. Depending on what type of hormone we're looking at, determines how in which it will activate signal transduction. So if we look at a steroid or a lipid hormone first, we can investigate how it initiates signal transduction. So first of all, a signal must be received. Then a message is created, and the messenger to send that message is the hormone. So then what needs to happen is that hormone needs to reach a target cell to elicit a response. So the steroid hormone has travelled through the bloodstream and is now in contact with its target cell. Because the phospholipid bilayer of this target cell is made of also a lipid, the steroid hormone can pass directly through the membrane. Inside the cell, the hormone will bind to a complementary receptor, creating a hormone receptor complex. In this particular case, the hormone receptor complex is going to move into the nuclear pore, interact with the DNA to initiate protein synthesis. Now, if we are looking at an amino acid or protein-based hormone, the way in which this type of hormone initiates signal transduction is slightly different. Now, the amino acid-based hormone has moved through the bloodstream and will find its target cell. The way it will recognise its target cell is that it will have a complementary receptor on the membrane. Now, with a steroid-based hormone, the receptor is within the cytoplasm, but because the protein hormone is unable to pass through the membrane because it's not a lipid, it has to bind to the receptor first, which is located on the membrane. A secondary messenger must be activated in order to carry the message within the cell. A really well-known secondary messenger is cyclic AMP, and what actually happens is when that hormone connects to its receptor on the membrane, there is something else embedded in the membrane called adenylate cyclase, which is an enzyme. Once the hormone has bound to the receptor, it initiates this adenylate cyclase to convert ATP into cyclic AMP, or little c, capital AMP. When cyclic AMP is activated, it can then go and initiate a response from another enzyme sitting within the cell, activating that enzyme into creating a metabolic pathway, ultimately creating a new response within the cell. Now, if we have a look at one of the examples in the Biozone book, we can actually see side by side the difference in which a protein hormone versus a steroid hormone activates signal transduction. So if we look straight away at the first one, hormone action by a secondary messenger, you see here that we have the peptide hormone, which is obviously an amino acid or protein-based hormone, bind to its receptor on the membrane, initiating adenylate cyclase that then converts ATP into cAMP. This then generates a response in protein kinase, which is inactive up until this point. Protein kinase is a type of enzyme. Therefore, we then get an alteration of the cell function through either a metabolic pathway starting or stopping. If we then look at gene activation by steroids, you can see here is a steroid that passes directly through the membrane and forms a hormone receptor complex within the cytosol. This hormone receptor complex then moves into the nucleus to activate DNA, altering the cell function by creating a new protein.